Okay. One more thing, the CoGuardian. All right. If you just came in, good afternoon. We're going to go to the module. Look at your to-do list on the module. And as soon as I get GoGuardian started, I will share with you. Okay, that's done. But I want to show you how to use the chart and the wheel, show you what's the difference between the two, and then give you a heads up on how to do the coding. So we were looking at making proteins. We were looking at DNA model, and we were looking at the protein and talking about the fact that our RNA has copied our information. So we are missing 2 people. Okay, well, hopefully they'll come along shortly. All righty, so let's share. Okay, so again, if you're looking at your home page, you'll see something that says under the to-do list, protein synthesis and code practice. And when you open it up, it looks like this. Okay, so you want to download it so that you can write on it. All right, I'm going to pull mine up because I believe I can't write on this one, but I can write. I saved the one that I did write on. Okay, give me a second. Okay, well, it didn't save my answers, but that's good too. So we will start afresh. All right, so let me change my share view so that you're looking at the one that I can write on. All right, that'll be fine. Okay. All right, so what we did last week, we learned the 2 words transcription and translation. And if you remember from last week, that transcription is going to happen in the nucleus with the DNA. So that's where the DNA unzips, the RNA lines up with its nucleotides and forms a copy of the DNA. And then the DNA zips back up and the new chain of RNA will leave the nucleus, go into the cytoplasm, and make its way to the ribosomes. At the ribosome, so now that's the messenger RNA. So at the ribosome, it enters like it's a template to be read. And the messenger RNA, the transfer RNA says, I want to make this protein, so I have to go and get it. So it's the transfer RNA is kind of a messenger, except there are little amino acids floating around and they are attached to the transfer RNA. Okay, just give me 1 second to turn off my phone. Okay. So it travels in the cytoplasm, gets the amino acid that it needs and brings it back and lines it up on the template. Maybe it might help if we kind of look at that again. Hold on 1 second. Just, I know over the weekend, you didn't think anything of this. Let's see. Oops. Okay. I have to come out of student view because then just to get a refresher on what's going on. I have to go back to change the viewing, but I'll do that as soon as I get to where I need to be. All right, while I'm doing that, anybody have trouble downloading the worksheet? So by now, everybody should have downloaded a worksheet, the one that looks like the one on the screen. And I'm going to walk you through how to do it, just trying to backtrack as to what it is we're doing, why we're doing it. Okay.
Step 2, translation. The purpose.
All right, this one didn't have any pictures. I did have one lined up for you that had pictures. So give me 1 second to pull that video up. I feel like you had a long weekend. You probably forgot everything we talked about last week. So we need a visual as to what it is we're doing. Let's try this one. It's short. I'm going to stop this for a minute and do this short one.
Want to break into cybersecurity but don't have experience? Physical cybersecurity uses real-world scenarios to teach you technical skills. You'll be able to step foot and feel comfortable and confident in the skill.
Protein synthesis, translation. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, mRNAs move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm is an enzyme, aminoacyl tRNA synthetase. The enzyme macromolecule has 2 binding sites. One site recognizes the amino acid methionine. This is followed by binding of the ATP molecule and release of pyrophosphate, resulting in activation of amino acid. Finally, tRNA and the activated amino acid bind together. This amino-acylated tRNA is known as met-tRNA and is released from the enzyme. This marks the commencement of the first stage of protein synthesis, the initiation stage. During the initiation stage, a small subunit of ribosome binds to the mRNA strand. The mRNA strand is made up of codons, which are sequences of 3 bases. Then the ribosome subunit moves along the mRNA in 5' to 3' direction until it recognizes the AUG codon or the initiation codon. At this point, met-tRNA, possessing the anticodon UAC, pairs up with the AUG codon of the mRNA. Then a large subunit of ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the aceptor site or the A site, the peptidyl site or the P site, the exit site or the E site. This whole unit forms the initiation complex. This is followed by the elongation stage. During this stage, another tRNA-carrying molecule of an amino acid approaches the mRNA ribosome complex and fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. As a result, met-tRNA becomes deacylated. The ribosome then advances a distance of 1 codon and the deacylated tRNA shifts to the E site from where it dissociates. Meanwhile, another tRNA-carrying amino acid molecule attaches to the A site. This is followed by the binding of the amino acid molecules. Repetition of this process leads to the formation of an amino acid chain. This event is called elongation. Finally, when the UAG codon or the stop codon reaches the A site, elongation is terminated. Termination is the last stage of protein synthesis. The chain of amino acid molecules is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation. Then the tRNA detaches from the mRNA. Ribosome detaches and dissociates into its small and large subunits. Summary. Protein synthesis shows that the first stage involves the binding of the met-tRNA to mRNA and the small subunit of the ribosome. The larger subunit of ribosome then combines with the small subunit. The second stage is the elongation stage. In this stage, the incoming amino acyl tRNA fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. The process is repeated until a chain of amino acid molecules is formed. The last stage of protein synthesis is the termination stage. When the ribosome reaches the stop codon UAG, elongation stops and the newly formed amino acid chain, which is the protein macromolecule, detaches from the ribosome. Subsequently, ribosomal subunits, along with the tRNA, dissociate from the mRNA.
Okay. All right, so that's in a nutshell what we're going to do today, and I'm going to go ahead and put this in the playlist for you. All right, so let's come back to what we're doing today, which is, let me Oh, I need to get back to the one I can write on. Okay, hold on. I got to stop sharing so that I can change share screens. Okay, there we go. All right, so everybody has their worksheet up that looks like this. So she said a couple of things. She's talked about a codon. Codon is 3 bases together, and each base codes for an amino acid. So every time the transfer RNA goes off, it's got 3 bases attached to it, and those 3 bases tell it which amino acid to go look for. Okay, so now on my right, you have 2 charts. I like the chart version better because I think it's easier to read, but the circle version is exactly the same, and I'll show you how to use both. All right, so the chart version, what we're going to do is take those 3 bases and we're going to decide what amino acid it's coding for, and you're going to put that on the line. There are 20, and then it's not on the worksheet, but in your notes, you need to remember there are only 20 different kinds of amino acids. Some of these, there are different ways to code for that amino acid. So in some places you'll see that there are 2 and sometimes 3 different ways to get that same amino acid. But this is how we're going to do that. Notice the box says first base, and at the top it says second base, and on the right hand side it says third base. That's how you're going to look at, for example, our first thing that we're looking at is CCC. So we're going to take the first one and we're going to sign C on the side that says first base, and we notice that it is the second row. Okay, so that's telling us which row we're going to end up in. All right, our second base is also a C, so we go up at the top and notice that C is in the second column. So now we know we can come down the second column to the second row, and now we need to look at, but there's 4 different messages in there. So we look on our third side, and C is we kind of have to go in line with the C on the third side, go all the way across, and then you'll see CCC. Now, just so happens in this case, there are 4 different ways to write proline. So what we're going to do on this line, come on, can I write? Is type P up. This is not letting me write. The whole reason. Anyway, on this line, you're going to, let me change the way I do this so I can edit. But you want to write PRO. PRO is short for proline. So you're going to put PRO on the first line. Let me come back out of this. Okay, hold on. Oh, it won't let me write on it. That was the whole reason for pulling this up. Okay, anyway, CPRO in the box up here at the top where we had CCC, that's what you're going to write on the line for CCC. You're going to write PRO, which is the short version of proline. All right, second one, and I'm going to do 3 of them for you, and then I'm going to switch down to the bottom one to show you how to do it in reverse, and then I'm going to show you how to do the same thing with the wheel. Okay, the second one is CAG. Again, we're going to start with C where it says first base. We're going to, and that's telling us it's in the second row. We're going to go up to the top for our second base, and that's an A, so that's our third column. So it's in the second row, third column, and our last letter is G, which is on the bottom. So if we're on the third, so we slide over to our third column, and we find CAG, and that's coding for GIN, which is short for I forget the full name of that, but you're going to write GIN on the line for that one. All right, let me show you how to do it on the wheel. So we're going to take GAA and we're going to look at it on the wheel. With the wheel, you start from the middle and you work your way out. So instead of having 3 sides, it has 3 circles. And actually it has 4 because by the time you get to the outer circle, you have the name of the amino acid. So if I start in the middle and I find G, so I know I'm dealing with that quadrant up at the top left hand. Our next letter is A. So right above G, I go my second circle. I'm looking at the A. And then the third letter is A again. So I go up and I look at where that A is and what the amino acid is in front of that A is GLU. Okay, so you're going to put GLU on that line for GAA. Okay. All right, so that's how you use, so use whichever one works best for you. If you like the box version, use the box version. If you like the circular one, use that one. They're both the same. They're both going to take you to the same endpoint. All right, so I did those 3 for you. You need to finish the rest. Now look down at question 2. I'm going to do the first 3 for you. So now this is doing it backwards. All right, so and they've bolded the first 3 letters of all the names because that's the only thing that's going to see on the chart. All right, so proline is PRO. So I look up at the chart and I see PRO, and that's in our second column, second row. And there are 4 possibilities for that amino acid. So I want you to write all 4 on the line. Okay. Then you take valine, VAL. So I look up at the box and I try to find where VAL is. VAL is in the G box, first column, last row. Okay, so again, there are 4 different possibilities. Please write all 4 possibilities of the way we can write valine or valine. Okay, histidine. So we're looking up at the box, and this is where the box is easier than the wheel. It is, if you go CAC, CAC or CAU, there's 2 versions, 2 ways to get that amino acid. So that's what you should write on the line. Okay, so at the top you're writing the amino acid. For number 2, you're writing the codon, the triplet codon for that particular amino acid. Okay, all right. On the next one, let me show you. So 3 and 4 are just answers that you need to find on the chart. All right, the first one, actually, let me help you with that too. 3 and 4, I'm giving you the answer. So the 3 wants to know where's the start codon. So if I go back over here on the chart, and she talked about it in the video, this is the codon that starts it. So MET, so if we look for A, go first column, third row down, AUG, and it says MET or start. So that's what you want to write in that for number 3. MET methionine is the amino acid that tells it to start making this particular chain of amino acids. Question 4 asks you for stop. Which one is stop? And here you have 3 different ways. So if you look at the top, in the orange section up at the top, so the yellow one is your start, and the orange one, there's 3 different ways to stop. So there's A is UAA, UAG, and UGA. So write those down for the stop codon. All 3 of those will tell the chain to stop making that particular chain of amino acids. Okay, so there's one that makes it start, and then as she showed you in the video, from there it will keep on bringing the answers to the code, and then it'll stop. All right, so that's 3 and 4. I am going to leave you something to do. Not giving all the answers away. All right. Actually, give me 1 second. I'm going to close this and try to. Oh, it stopped sharing. Okay, give me 1 second to come back. I'm going to show you how to do the other question. Okay, that was not it. I saved one with answers on it. I thought I saved it. So I wouldn't have to keep typing this over again. Okay. And it came back up the same way. Okay. It's not working with me today. Alrighty. So that takes care of those. All right, so I've answered 3 and 4. Now let's look at 5. For 5, you've got 2 rows that you have to complete. The first row, you're going to have to build the RNA strand, which means you have to remember who matches with who. Okay, and you have to remember also that RNA does not have T. So while T will bond with A, and it's making me crazy that I can't write on this. Okay. All right. So T, you want to match that up with A, but A, you want to match with U, and then C with G. And you want to go across this whole line and do your complementary triplets. Then you want to go back after you've created your complementary triplets. Then you want to go figure out what amino acid am I looking for. So you want to go, so with this, it's going to end up being AUG. So we're going to go back to the chart and we're going to look up what is AUG, and AUG is our methionine. So that's what you're going to write. Where it says amino acid, you're going to write methionine. And remember, that's our start codon, so that works. And then you want to go across the whole thing. And for every triplet, you want to come back to the chart and find the amino acids you just coded for. Okay, so you've got 3 rows of that to do. And then you're finished. Okay. Now, if you don't code right, you end up with a mutation. So it does matter. It will collect the wrong amino acid, and then you end up with a mutation. So take your time and write yourself a key that what the RNA will be. Remember, there is no, write down for your information for yourself that T is going to match A is going to match U, G is going to match, C is going to match G. And that will get you through all of those. So you're creating a row of RNA, and then you're creating a row of amino acids. All right. Everybody good? Any questions? Okay. All right, so fill in all the ones that I didn't do and then fill in these question 5. You have 3 rows in question 5. I'm going to stop sharing so I can do attendance, but feel free to ask questions as needed. All right, Michaela, this worksheet was on your to-do list on your on your module page. LaCoria, you too. All right, so this shouldn't take you long. You should be able to finish this one today and upload this. So this is a today assignment, not a let it hang over till next class assignment. All right, so it looks like right now the only person we're missing is Jacob. Again, this worksheet is due today. It shouldn't take you long. If you get finished before the end of class, go to the, in fact, this chart is also in your, on the next page with mutations. The chart that we're looking at, those 2 charts are listed there as well. All right, everybody good? Everybody understand what they have to do with the worksheet? So first set, you're looking for the amino acids. For the amino acid, second set, you're looking for the codon that would code for that amino acids. If there's more than one, write all of them. Then we did 3 and 4, the amino acid for start, and there's 3 of them for stop. And then you're doing 3 chains of creating RNA and then finding the amino acid that is being coded for. All right, LaCoria, you there? I'm looking at your ceiling or someplace, but I don't see. Okay. Lovely ceiling. And again, the thing to remember is it's a triplet codon. For every 3 nucleotides, for every 3 bases, there is There is one amino acid associated for every 3 bases. It doesn't matter if you use the chart or the circle, you should come out with this. It should give you the same amino acid. And I was just about to pull up uh, Canvas to show you the same chart is in Canvas charts. All right. So in Canvas on your page, it says mutations lesson. You have the same 2 charts there as well. And we'll talk about mutations if we have time. I think, well, this will probably take you to the end of the lesson. So on Wednesday, we'll talk about mutations. And mutations just come if you read the code wrong and you pick up the wrong amino acid, it makes a wrong protein. And the proteins are designed to be part of a structure. So if you're making an eye, you want it to have all the right proteins, right amino acids to make the protein for the eye. If you put in a wrong amino acid, there might be a defect. Sometimes you can survive a mutation, sometimes mutations can be lethal. So reading this chart is very important. All right, the nice thing about the one on Canvas is it has all of the names of the enzymes spelled out, not just the first 3 letters. But typically you'll only see the first 3 letters on a chart. They can be abbreviated. All right. I had the chart does look like it says GIN, but it's GLN. So it's not GIN, it's GLN. Okay. It's kind of hard to read because it's so small, but that's GLN for um, glutamine. Again, if you're finished with this assignment and have uploaded it, read the mutations page. I'm about to add an activity, but I don't expect you to get to it till um, Wednesday, but I'm about to add an activity for practicing mutations. All right, so don't forget that the objective was to try to get this finished today so that we can do something else on Wednesday, but it's not late until Wednesday, but try to make sure that it's finished at the beginning of class on Wednesday because we do have another assignment for Wednesday. So on Wednesday, we're going to look at doing the same thing, except now looking at a mutation. And a popular mutation is sickle cell anemia. So that's the one we're going to look at. Okay, if you have no questions for me, you're free to go, and I'll see you all on Wednesday.